All right, so the evolution of drug-resistant bacteria is a good example of microevolution, and also illustrates the points of how natural selection occurs. So uh, one common antibiotic-resistant bacteria that we've heard about a lot is the MRSA, as it's pronounced. It's a type of staph bacteria. The full species name, Staphylococcus aureus, is a common bacteria. It's probably everybody has some on their skin if we swabbed you. Only some strains are bad. Um, and the MRSA is an acronym, and it could stand for methicillin-resistant Staphylococcus aureus, and it also could stand for multiply resistant Staphylococcus aureus, because some of them are resistant to more than one antibiotic. Some bacteria have been found that are resistant to up to 6 different kinds of antibiotics. And pretty much every time a new antibiotic hits the market, very quickly, in a year or two, the first antibiotic-resistant strains to that particular antibiotic show up. And this was true even from the very first widely used antibiotic, penicillin, which started to be widely used at the end of World War II. And it became the first antibiotic-resistant bacterial strains were discovered not long after that. So what exactly happens when a bacteria becomes antibiotic resistant? So bacteria exist in extremely large populations, millions and millions of individuals, and their mutation rate is higher than in eukaryotic cells, like in our cells. They reproduce much more quickly. Every time their DNA replicates, mistakes are made. So new mutations are occurring randomly all the time, every generation. Every time a cell divides, there are new mutations. So if you have a population of bacteria, just like any population, there's variation. A population, if you have a staph infection in your skin, that population varies in its genetic makeup. And it's possible that a few of those cells in your of the bacterial cells are going to have a mutation that will allow them to survive an antibiotic. Now, normally, that's not a big problem, because the way antibiotics work is they don't kill every single bacterial cell. They rely on your immune system to be part of that process. So what antibiotics do is either stop the reproduction of the bacteria or slow it down, or kill most of the bacterial cells, and then your immune system mops up the rest. If you have a non-functional immune system, antibiotics will not help you because they rely on the immune system as being part of the defense process against bacteria. So in a healthy person who has a sore throat or a staph skin infection, they give you antibiotics. The antibiotics slow the replication rate or wipe out most of the bacteria. Your immune system cleans up the rest. All is good. But in a sicker person, somebody who has a poor immune system, if they have an infection that has a few individuals that just by chance have a mutation that will allow them to be unaffected by the antibiotic, we give an antibiotic. Now, their immune system is not going to wipe out the remaining population. Instead, that population is going to replicate, and now the infection that they have is with a bacterial strain that's mostly resistant. Now, if this strain ends up infecting other hospital patients, now we have a case like has happened in many hospitals, where you end up with multiple patients who have the same antibiotic-resistant bacterial strain, sometimes resistant to multiple antibiotics, being passed around the hospital, carried on equipment, carried on the clothing of staff in the hospital, and unknowingly transmitting the infection from patient to patient. So that's why these antibiotic-resistant strains so often show up in hospitals, because you have a concentration of people who have a poorly functioning immune system. They get an infection, they're given an antibiotic, and if there are mutations present for some kind of antibiotic resistance, they may end up then with a recurring infection that is now antibiotic resistant. So notice the mutation was present before the antibiotic was used. The mutations are just occurring randomly. So in a given bacterial population of millions of cells, there's going to be a sprinkling of individuals that have mutations that will allow them to survive different types of antibiotics, just randomly. If we happen to give an antibiotic, now that is a selection event. Now there's our natural selection right there. So now we have variation in the population. We provide a selective event, high death rate of most of the population, and now the surviving population is going to be the descendants of those resistant cells. So the antibiotic doesn't cause mutations to appear, it only selects which individuals are going to survive to reproduce. Another topic that is often addressed is how do very complex things evolve, especially things that don't really fossilize, like the structure of eyes. Our eyes are very complex. We have a lens that can accommodate for distance. We have a living but transparent cornea. We have an iris that can open and close and accommodate for different light levels. How could something this complex evolve by mutations and natural selection? So someone, geneticist, tried to calculate how many independent positive mutations it would take to go from the simplest kind of eye in animals, which would be just a layer of cells that can respond to light, to a complex camera-like eye like what we have, and they counted about 300 mutations would be needed. And if you have millions of years to work with, I mean, animals have been evolving since the Cambrian explosion. That's plenty of time for mutations that are needed to occur. Some people have argued that our eye is poorly designed, and that if you were an intelligent designer designing our eye, you would not design it the way that we have. And we see situations like this all the time in organisms where you think, okay, that's very bad engineering, but natural selection only has randomly occurring mutations to work with, to select for. So the solution that occurs isn't always what you could think of as the ideal solution. So in vertebrate retina, we have the rods and the cones are at the back of the retina. Those connect to neurons, which are going to go and connect to the optic center of the brain. And the cell bodies, so you see the nuclei here, these are the cell bodies of the neurons. They're actually in the way of the path of the light. So they dim the amount of light coming in to the rods and the cones, the photoreceptor cells, because they're in the way. And this seems like a bad idea. And it also leads to a blind spot in our retina, because all of these axons here of these nerves of these neurons are going to collect together and then dive out of the eye through the optic nerve. And so this means that there's actually a hole in the back of our retina. There's a blind spot. And if you look straight ahead, you have a blind spot. If you think of your visual field as sort of a clock face, right around 8 o'clock and 4 o'clock is where your blind spots are in your eyes due to this. And this, seems like a bad idea. Why don't we see a black circle where the blind spot is? It's because our brain fills in with whatever's around that spot. So we don't see our blind spot. One of the ways we can try to understand how these soft structures evolved that have a poor fossil record. So as eyes themselves don't have any hard parts, so they don't fossilize very well. So trying to understand the evolution of something like this, we can look at organisms that have different types of eyes. So they're at different stages of the evolution of the eye, and that can help us understand this. This is what good is half an eye? This is a tagline from a particular intelligent design book that argues that our eye is too complex to have evolved by natural selection. But vertebrate eyes are pretty much all the same. If I go back a slide, our eye is very similar to all the vertebrates. But if we look at mollusks, we see a lot more variation in the type of eyes in living mollusks. So we can't rely on fossil mollusks. So mollusks are everything with a shell that includes snails and clams and oysters, but also octopus, nautilus, squid, snails. Those are all mollusks. And mollusks have a huge variety in their types of eyes, much more so than vertebrates do. And if we look at the simplest kind of eye, we find living mollusks that have this type of eye that's just a simple flat sheet of photoreceptor cells that are connected to neurons. And all they can tell, each cell can only tell, is there light? Is it dark? And this is useful both for telling the difference between day and night, but also if a predator swims over and it suddenly gets dark, they can close their shell. The next stage in eye evolution would be a simple cup shape that kind of protects these photoreceptor cells from abrasion, and there are mollusks with this type of eye. A simple cup shape would be the next type, and there are living mollusks that have this type of eye as well. It's still open to the sea, but it's more protected against abrasion. No ability to focus, no ability to accommodate for light. The next type of eye here, which we find in marine snails, has a refractive lens which allows them to focus better, but no accommodation for distance or for light. Now the photoreceptor cells are much more protected. They have a transparent epithelial layer that allows light to go in, but very low ability to focus on things, but better than this eye. So there are, living mollusks with this kind of eye. Now the most complex kind of eye, very similar to our own eye, is found in octopus and squid and cuttlefish. And this has very similar features to our own eye, a lens that accommodates for distance so they can focus crisp images on things at a distant and close up, transparent living cornea, a retina, an iris that opens and closes for different amounts of light. So very similar to our own eye, but we know that this evolved completely separately, that the similarities we see are the result of convergent evolution. This eye is analogous to our eye, not homologous. So our common ancestor with mollusks had an eye like this. So how can we tell that this is analogous? It's because the details are different. So if we look at the details, it's different. For example, their optic nerves and all these neurons that connect to the photoreceptor cells in the retina are all behind the retina, not in front of the retina like in vertebrate eyes. So when the details are different, that tells us that structures evolved independently and that they are analogous, not homologous. Another criticism is this is another one that comes from the intelligent design community, and they call some things irreducible complexity. Just like the evolution of the eye, they say it's too complex to have evolved by mutation and natural selection. They say the same thing about the blood clotting system. So you might know that human blood clotting and other mammal blood clotting is a fairly complex system involving more than 13 different proteins. And there's several proteins that interact together to create the clotting system that we have. And this also doesn't fossilize. So how extinct organisms clotted their blood, we probably won't know. We can't get their DNA if their fossil remains are more than a few thousand years old. So how can we know how a complex system like this evolved? Well, we can, again, just like with eyes, study organisms that are alive today and ask what parts of the clotting system do they have. And in fact, if you look at vertebrates, fish to humans, you can see that they have different parts of the clotting system. So the oldest part of our clotting system turns out to be the actual scab-forming proteins or clot-forming proteins. Thrombin and fibrin, which you probably heard of. We have a protein that prothrombin that's cleaved to form thrombin when there's an injury, and fibrinogen, which is cleaved to form fibrin. And these form the structure of the clot. Even the most primitive vertebrates have this system. So the most primitive vertebrates are the jawless fish. They have no bony lower jaw. Instead, they just have a soft, kind of worm-like mouth. Those are the jawless fishes. Hagfish are jawless fishes. Lampreys that cling on to sharks are jawless fishes. They only have this part, which is called the common pathway. Don't memorize the terms for this. This is just an example of how we can study how a complex system evolved by studying living things today. Okay, so the most primitive vertebrates, they only have this. They still manage to clot their blood just fine. Now, the jawed fishes, this includes all the bony fishes, sharks, and goldfish, and tuna, and all the common fish. They also use another pathway called the extrinsic pathway, which we have too. And a lot of the proteins that are involved with blood clotting have Roman numerals associated with them. So factor 7, factor 10, factor 5. Reptiles have an additional system, the intrinsic pathway. We have all of these. Reptiles have the intrinsic pathway, so we know this evolved about 450 million years ago. And again, we see a few more of these protein factors. Each one of these, factor 10, factor 8, factor 9, these are all proteins that interact with each other as part of the reaction to injury to form clots and scabs. Factor 8 we're going to talk more about. This is the one that's involved with the most common type of human hemophilia because it's the only one that's on the X chromosome. So we're going to talk more about that when we get to genetics. So, and finally, one more factor, factor 13, which adds to fibrin to create a cross-linked clot. This one is in mammals. So mammals have all 4 of these. So this is, in fact, a very complex system. And in humans, there are clotting disorders that are known if an individual is missing any one of these factors genetically because of mutation. They're going to have problems clotting their blood. So that's true that in humans you can't be missing any one of these or you're going to have some kind of a blood clotting disorder. But that doesn't mean that we can't understand how a complex system like this could evolve. So the fact that blood clotting doesn't fossilize, the way that we would study a system like that would be by studying animals that are alive today and asking what variation do we see when we compare greatly different groups of animals. So we talked about in this chapter that Darwin got some of his ideas, the fact that evolution is occurring, that species are changing over time. He got that idea from earlier scientists, from Lamarck, from his own grandfather who thought species changed over time, but he came up with a mechanism. He was the first person to come up with the mechanism that we call natural selection, which he called descent with modification. And there was also other things that he came up with as well that he was the first person to come up with. He was the first person to say that all life on Earth is related by descent and that we could trace back all life on Earth to some first cells that were the original life forms. He also was the first person to say that chimpanzees are our closest living relative. And he based that, there were no intermediate fossils known at the time. He based that entirely on studying morphology of chimpanzees and observing the couple of captive specimens that were in Britain at the time. Natural selection is what we now call descent with modification, meaning that some individuals are more likely to survive to reproduce based on the environment. He waited many years to publish until Alfred Russel Wallace came up with the exact same mechanism. We talked about his direct observations. There's variation in a population of heritable characteristics. And he knew from his conversations with farmers and animal breeders that new traits appeared, and those new traits were often heritable. Organisms produce more offspring than the environment can support, so there is individuals who don't survive every generation. So what he inferred from that is that, well, if we have variation and excessive production of offspring, then every generation, the individuals that have better adaptations to the environment are going to survive to reproduce, or reproduce more than other individuals. And that's going to change the population. And new traits that appear that are favorable would mean that it helps that individual survive and reproduce. Those traits are going to accumulate and become abundant in a population, and other traits that are less favorable are going to die out. We talked about the evidence for evolution, the fossil record, vestigial organs, which are even found in humans, pseudogenes, which we could also call vestigial genes, genes that are inactivated but were used in an ancestor, vestigial structures and vestigial organs. These are things that are useless in the organism it currently is in, but were useful in an ancestor. Homologous structures versus analogous structures. So homologous structures are inherited from a common ancestor. If they're analogous, it means they're the result of convergent evolution, and they look similar and they serve the same function, but they were not inherited from a common ancestor. Like the dorsal fin of ichthyosaurs and dolphins is due to convergent evolution. And we now can compare DNA as well to determine similarities between organisms.